Okay, that worked. And so we'll make you the host again. Okay, so that should be good. Okay, well, good morning, good afternoon. Thank you for coming. Welcome to our kickoff Dementia Journal Club for the 2021-2022 school year. Today, I'm broadcasting live from Nova Southeastern University campus in Fort Lauderdale, Florida. So I want to say hello, a big hello to my friends in Canada. Of course, there's been a big change in my location. As I asked a few seconds ago, if you wouldn't mind just turning off your microphones for now, we hopefully will have time at the end for a few questions. But today I have the pleasure of introducing Dr. Richard Deth, who joined Nova Southeastern University in 2014 after he was already at Northeastern for 38 years as a faculty member, a professor, and also former chairman at Northeastern up in Massachusetts. I hope I have all that right. So here, Dr. Deth is a professor and the founding director of the Brain Center at Nova, which stands for Brain Research and Integrated Nutrition. His expertise is as a molecular neuroscientist, and his laboratory focuses on mechanisms underlying neurodevelopment, changes that occur during aging, and also mechanisms involved in attention and learning. So in particular, he's looked at factors influencing the levels of the antioxidant glutathione, which many of you know about, and its influence over methylation reactions, including DNA methylation. Dr. Deth has also worked closely with autism families and several patient-supported autism organizations. His current research involves studies on the effects of neurotoxic substances on antioxidant and methylation status and autism-related changes in antioxidant and methylation, as well as vitamin B12 levels in post-mortem human brain tissue. And today he's going to present a paper on redox regulation in the NLRP3 inflammasome. So I'm going to turn it over to you, Dr. Deth. Thank you very much.
Thank you, Ben. I appreciate the opportunity to speak today and to people who know more about Alzheimer's than I do. But being a newcomer, I can say whatever I want. And I can try to leverage my background and information to make it useful and maybe even interesting to people here today. So you can see from the title, I'm going to talk about the NLRP3 inflammasome and related to Alzheimer's, and especially focusing on the redox aspects of that. And let's see here, the overall plan here, I got different sections here. You'll see them as they come along. We'll talk about redox first to make sure everybody's on the same page, or on my page at least, and then we'll relate it to what's going on in Alzheimer's, then move on to the NLRP3 itself, and wrap up with some thoughts about treatment. Let's just start with what we know, particular, I know, I guess, about the aging brain, mainly from our postmortem studies, but from other people in the literature as well. Just by way of general background, this is what everybody knows, but I'll put a slide up here so we agree on it. That the brain uses a ton of oxygen, uses 10 times as much as any other tissue. And as that occurs, it obviously is going to be an issue for being able to safely use so much oxygen. And ultimately, it needs to match the antioxidant demand with the antioxidant supply. And the antioxidant glutathione plays a major role in providing that antioxidant resource. And in particular, it's evolved in the brain to play a regulatory role, as well as simply being like a bulk antioxidant. And of course, the basis for the high energy demand comes from the firing activity of neurons and electrical activity that requires ATP. The glutathione molecule, for those who don't think about it all the time, it's a tripeptide. And so it's got a gamma, it's got a glutamate linked through its side chain here, and to cysteine, the sulfur amino acid cysteine, and the SH group in the cysteine, it's the functional heart of the glutathione. It makes it what it is. And there's a glycine on the other side here. And what's limiting for making and having available glutathione is the cysteine. It's the limiting factor. And so the uptake and the availability of cysteine is critical for the brain, as well as the net amount of glutathione. And the neurons have the least amount of glutathione of any cell type in the body, despite the fact that they probably have close to the highest need for glutathione. And so this makes oxidative stress a possibility for neurons, and hence the danger associated with neurodegenerative disorders. So this is the diagram here, a metabolic pathway diagram that our lab uses, and we've used and developed it and enhanced it over the years. Probably has to undergo some further revision, as always. But what you're looking at here is trying to capture the pathways that make glutathione in neurons. And this would be the red pathway with glutathione or GSH at the top. And the supply of cysteine for that synthesis of glutathione comes from cysteine uptake that's gated or otherwise stimulated by neurotrophic growth factors via the E8T3 transporter here. I'll talk later on about how that activity is inhibited by the A-beta peptide. But the cysteine that's taken up is derived from astrocytes. And astrocytes release some glutathione. They have about 4 times as much as neurons have. And they release some, and it breaks down to cysteine that's available for neuronal uptake. So this is a major source of cysteine for glutathione synthesis, especially when you're young. But there's an alternative pathway that gets more important with age. And that's the one that derives from the methionine cycle of methylation that's in black here on the lower right. Because when the homocysteine is generated from methylation reactions, the homocysteine can move through transsulfuration, this 2-step pathway here, to be turned into cysteine for glutathione synthesis. So you can get the cysteine either from outside or from inside the cell, if you will. And the decision about how much homocysteine will be diverted to transsulfuration is largely controlled by the enzyme methionine synthase, probably our favorite enzyme. And this is the vitamin B12 and methylfolate dependent enzyme that otherwise recommits homocysteine to the methylation cycle by putting a folate-derived methyl group onto the homocysteine. And so that pathway dictates whether the homocysteine will go north in this diagram or south to support methylation. The enzyme methionine synthase is really sensitive to oxidative stress. Matter of fact, the cobalt cofactor, the B12 cofactors, is the most easily oxidized molecule in the body, and therefore becomes a sensor for oxidative redox state in the cell. In blue here, I'm not going to talk about that at all today, is what got us started in this sort of journey in the sulfur metabolism. That's a D4 dopamine receptor that carries out a unique methylation reaction involved in attention here. But what got us more in the direction of Alzheimer's was the fact that we measured the gene expression in postmortem human frontal cortex of that methionine synthase enzyme. And that's what you see here on the left. And we measured it in samples from normal subjects across the lifespan, from really prenatal fetal levels down to 84 years of age. And you can see a rather remarkable and normal pattern of decrease in the RNA level, messenger RNA level, for this enzyme in the frontal cortex. It goes down really steeply until about mid-teens or something like that. And then it continues to go down more gradually during adulthood. And we thought that was kind of remarkable. It's about a 400-fold decrease by the time you get to be 80 years old, and that's a lot. We also had occasion to have samples from individuals with autism. They, in the right-hand side here, you can see they all had almost all lower levels of methionine synthase message than did the age comparable peers here, suggesting that there was a relationship to the origins of this neurodevelopmental disorder, autism. So in any case, we became interested in the aging effects on these sulfur pathways here. One of the other things that we discovered, if you will, at the same time, was that there's alternative splicing of the messenger RNA, which is depicted here on the left for methionine synthase. And that alternative splicing removes a portion of the protein called the cap domain, shown in yellow here. And that cap domain normally protects against oxidative inactivation of the enzyme. And when you get older, that cap domain disappears. You can sort of see that here. 24-year-old had a cap domain band, but the 80-year-old samples here didn't. And on the average, you can see a decrease with age that's rather impressive. And so this was a second way in which the methionine synthase enzyme activity decreases with age, in this case because of a alternative splicing of the messenger RNA. And then we went on to measure the brain levels of the cobalamin cofactor, which is methylcobalamin or methyl B12 here. And on the right side, you can see the results of an age study here, 0 to 20 years, 20 to 40. It's about the same level of methyl B12, but starting from 40 to 60 and then 60 to 80 years, there's a rather steep fall off in the levels of the cofactor for that same enzyme. This means, together with the other observations, that it looks like there's a decrease in the support for the enzyme methionine synthase with age, especially advancing age here. We also had a chance to measure the metabolite levels. Homocysteine is the substrate for that enzyme. And in the older brain samples here, 60 to 80, you can see the homocysteine level was high. And so this is maybe not a surprise that homocysteine goes up with age, but here in the brain, at least we can see how much that increase is. And the products of the reaction, methionine and SAM, are lower. So what this means is that there's a decrease in methylation in the frontal cortex of normal subjects here with age. This looks to be an adaptive response, a normal response that helps encourage transsulfuration at the cost of methylation. So in this illustration here on the left, again, is the methylation cycle, and its diversion of the homocysteine through transsulfuration goes up with age, whereas methylation decreases with age. There's a lot of ramifications to that, and we'll see some of them as we go through here today. But this transition is really important in terms of maintaining enough antioxidant, especially in older age here. And we wondered what might be causing this transition, the shift toward transsulfuration away from methylation. We took a stab at it, and we looked at the effects of TNF-alpha, the pro-inflammatory cytokine, and found that TNF-alpha decreased the cysteine uptake rather promptly in cultured neuronal cells here. It decreased the expression of the B12 enzyme, again, the methionine synthase, very promptly. This goes from 300 down to less than 30 in a half hour, something like that. It's a pretty dynamic decrease. And but the levels of the glutathione shown over here on the left in green, and the cysteine in black, they didn't go down. There was a transient increase, and they maintained their levels despite the fact that the uptake of cysteine was greatly decreased. This means that the transsulfuration pathway was was being activated and compensating for a decrease in the uptake of the cysteine. And this is that switch that I was referring to, a switch toward transsulfuration. And it implied that inflammation might be part of what's triggering that in the case of the elderly subjects that we looked at. So with that kind of overview of antioxidant and methylation sulfur pathways here, let's just look at how this plays out in Alzheimer's. Again, this is an area that you may be familiar with the background in Alzheimer's as much as certainly I am, but just to put this into place here, for example, a study in Sweden that was done in basically cognitively normal individuals over time, just looking at the prospects of developing Alzheimer's disease. They found that there was a relatively higher probability, depending upon the levels of homocysteine. If they were higher, that increased the risk. And if the levels of methionine were lower, that increased the risk. Well, this points exactly to that same enzyme, methionine synthase, as being an important factor for risk of Alzheimer's disease here. And they also found that the cystathionine, if that was elevated, that was associated with an increased risk as well. So in any case, there's evidence that these things are involved in the risk of Alzheimer's. They also looked at the correlation with the levels of vitamin B12 and folate levels here, and found that ratio was not unexpectedly was associated, was correlated with that methionine and homocysteine ratio there. So, but otherwise, the postmortem brain studies that have been done, this one in particular showed that oxidative stress goes up. Again, this is not a surprise in the frontal cortex with the progression of Alzheimer's from MCI, mild cognitive impairment, through full-blown AD here. And on the right, that the glutathione level went down. This is common knowledge, but this is just to say that this is exactly what occurs in the frontal cortex, where we were measuring those same changes in gene expression. The TNF alpha level goes up in the elderly, that's shown here on the left, and in mild Alzheimer's, full-blown Alzheimer's, it goes up further than the normal controls. And again, on the right, this was associated with a decrease in the glutathione level. So there's a reciprocal relationship between the glutathione level and the level of inflammation here, at least the TNF alpha level. So these kind of things really suggest that there's a link to Alzheimer's. And another aspect that we had a finger in, we did some studies on the A beta peptide, again on cultured neuronal cells, and as shown here, the A beta peptide inhibited the uptake. As shown here on the left on the bottom, this was the cysteine uptake without the A beta being added, and this was the A beta, which happened to be the supernatant from conditioned cells that were excreting the A beta peptide. You can see the A beta inhibits the cysteine uptake. It decreases the glutathione reduced versus oxidized ratio. It inhibits methylation. And over on the far right here, it inhibited DNA methylation, because all methylation reactions respond to the methionine synthase activity, which governs the SAM-SAH ratio there. Just to make sure people appreciate how that methylation decrease plays out, the phosphorylation of tau, or the hyperphosphorylation of tau, reflects, the putting on of phosphate groups, if you will, from GSK3 beta and other kinases, but the taking off, removal of them is controlled by protein phosphatase 2A, PP2A, whose activity depends on its methylation status. So PP2A gets methylated to be more active, and if it's not fully methylated, it's less active. So any impairment of methylation will favor hyperphosphorylated tau and neurofibrillary tangles and so forth. And again, in postmortem brain, people have measured the methylation status of that PP2A, and is shown on the left as a progression of Alzheimer's disease. Again, the methylation status goes down, indicating impaired methylation can affect the tau phosphorylation status. And the ratio of the methylated to the unmethylated PP2A, again, is down in Alzheimer's, but also progressive supranuclear palsy, or PSP. So another player here that we can now include is the hydrogen sulfide formation from this pathway, from the transsulfuration pathway. It's shown in the top here, the two enzymes in the transsulfuration pathway, especially in astrocytes, but also in neurons. The two enzymes, the first one, cystathionine beta synthase, the second one, cystathionase, or CSE, they both have the ability to produce hydrogen sulfide gas from either homocysteine or from cysteine. And because it's a gas, if the gas is produced in the astrocytes, which is the primary source when you're younger, the production of that gas in astrocytes can leak out, and the gas, hydrogen sulfide, can affect neighboring neurons, modulating their sulfur status and thiol status. When you're young, the CBS activity in astrocytes is really by far the dominant source of hydrogen sulfide. But with aging, the neurons start to produce more from the CSE enzyme, the second enzyme in the transsulfuration pathway. But overall, the hydrogen sulfide exerts antioxidant or neuroprotective effects that are beneficial. But in subjects, again, with Alzheimer's disease, the level of hydrogen sulfide in postmortem brain samples is low, less than half of what it was in the controls in this particular study. So again, we can see, and I'm sort of beating this to death, I suppose, but there's not only oxidative stress in Alzheimer's brain, but there's impaired methylation as well as low hydrogen sulfide. So there's a dysfunctional status of these pathways here. The recent paper from the lab of Sal Snyder and Bindu Paul at Johns Hopkins really painted an elegant story about the hydrogen sulfide's effect. The hydrogen sulfide actually modifies the GSK3 beta, therefore, and this has a net effect of decreasing the phosphorylation of tau. If you don't have enough of that, then you'll get hyperphosphorylation of tau. And as a part of that study, on the lower left over here, they measured the expression of that CSE, or cystathionase enzyme, in as a protein level here in AD patients, and you can see it was lower. They really had lower activity of the enzyme that produces the hydrogen sulfide. So this sort of also confirmed that. And so we did some studies more recently ourselves of these enzymes. On the right side here is that CSE, and we found too that the AD subjects had lower expression of the mRNA for the cystathionase enzyme compared to their age-matched older subjects there. And both of them were lower than the young individuals at least what we found. The other enzyme that makes hydrogen sulfide, the CBS enzyme, that showed an increase with age in normal subjects, and that was not different in the Alzheimer's subjects. So it looks like, based on that, that the Alzheimer's difference arises more from the CSE enzyme than the CBS enzyme. But then on the left, here's our introduction to the NLRP3, which stands for inflammasome, that we're going to talk about next. One of the inflammasome family proteins, the NLRP3 gene expression went up with age, kind of considerably here. And this is in frontal cortex again. But notably here, the Alzheimer's subjects had only a little increase above the young subjects. They were highly significantly lower in the NLRP3 gene expression, not the protein, but the gene expression. And this got our interest in the NLRP3 going a little bit more. So I'm not sure if everybody's followed the NLRP3 story at all, or some, or a lot, but it's a worthwhile thing to know about. I got interested in it myself really when COVID started, because it became clear that cytokine storm or runaway inflammation was what was killing people with severe COVID. And it was strange to me that some people had severe COVID reactions and other people had nothing, even though they had an infection. It didn't result in a serious life-threatening illness. So there must be some factors that are different in terms of the likelihood of having the cytokine storm and inflammation. And I thought that maybe the research that I knew about and that we were doing, especially with regard to autism, might have some relevance here to this story. So that's why I picked up an interest in NLRP3 here, and I'm still sort of pursuing that in an ongoing kind of way here. But let me introduce that here. The NLRP3 is a single gene, a single protein, whose structure, overall domain structure, is shown on the left. It's got a pyrin domain, a NOX domain, and a leucine-rich repeat domain here that sort of looks like candy or something like that. This NLRP3 is one of a family member. There's a lot of other NLRP3 proteins, and each one has some specificity. And it looks like the specificity for sensing something or responding for the NLRP3 is oxidative stress and glutathione-related status of the cell. And we'll see how that evolves as we move along here. But the NLRP3 is part of what's called an inflammasome. An inflammasome can be a very large structure. As you can see on the right side, a single NLRP3 molecule here, the NOX domain is in purple, and green is the pyrin domain, and then sort of turquoise is the leucine-rich repeat. This protein can bind another protein called NEK7, NEK7 here. And when it does, it can form this oligomer here, which is, in this case, has 11 different NLRP3 molecules joined together by their pyrin domain, which is what's holding them together. And once it forms, it's able to bind procaspase and initiate caspase activity that leads to the formation of cytokines here. So what I just described here typically is a pathway of cytokine and inflammation activation that is initiated by exposure to something that's either a danger signal or a damage signal, a DAMP, or a PAMP, a pathogen-associated molecular pattern here. And either of these signals activates through TLR receptors, typically NF-kappa B, as a transcription factor and turns on the transcription of genes for the various proteins, some of them I just mentioned here before, the NLRP3, the ASC protein, which is here on the previous slide, as well as the pro forms of IL-1 beta and IL-18, which have to be cleaved by caspase activity in order to release the cytokines here. So once the gene is turned on, that's the first step, the priming step, they call it. So the proteins are made and they're available. But a second step of the assembled proteins is the activation step. And that's going to determine the ongoing activity. This is the step 2, where if it's not turned off, it could be a runaway inflammation or a cytokine storm. So it's the regulation of the formed inflammasomes that becomes sort of critical for the final outcome of this inflammatory pathway here. Also on this picture, gasdermin D is another protein that's cleaved by the caspase activity, and that forms channels in cells. Ultimately, if the channels get big enough and numerous enough, cell dies, essentially from these pores that are in the membrane. Of course, they also allow the cytokines to come out and affect other cells. But the activation or the activity of the inflammasome is determined by a number of triggering factors. Oxidative stress for the NLRP3 is a prime trigger for keeping it active and making it active in the first place, but sustaining its activity over time. So we need inflammation. We want to kill that virus and otherwise activate the immune system, but we don't want it to go on when it's no longer needed. And that's where the problems come into play, is by not being able to terminate, if you will, or regulate the inflammasome activity longer term. Other triggers, a decrease in potassium inside the cell, that is potassium efflux, or the presence of extracellular ATP. If you're in the lab, you add ATP, it increases the activity of the inflammasome. And some other factors have been identified here that we'll run across that regulate the activity of the NLRP3. Just in passing, say, okay, this is involved with COVID. The COVID, or at least the SARS-CoV-2 proteins activate the NLRP3. This is in the literature and sort of agreed upon. And it's also agreed upon that the NLRP3 activity is what is responsible for a lot of the mortality associated with COVID-19 here. And some, if you care to look into it, and this study did here, you can identify visually the inflammasomes. They appear as what are called specks. Typically, one speck per cell can be visualized if you use, let's say, an antibody, typically against the ASC component. You can find inflammasomes in this case, the lung tissue of people who died from COVID. These specks here are apparent, and they are big enough to be seen by eye. So these are big. They're not just 11 molecules. They're 100, maybe 1,000 molecules, but they all aggregate to form one speck. So aggregation is the key feature of these inflammasome proteins. And their tendency to aggregate makes them remain in the cell and not be broken down by ubiquitination and or by other clearance of protein mechanisms here. So there have been some studies of the NLRP3 in Alzheimer's here. I suppose on the right side here, we can see the level of the NLRP3 protein, and this is in monocytes in white blood cells from individuals that are healthy controls, MCI, mild AD, or full-blown AD, and it's going up. So we can see that there's an association between stages of Alzheimer's and the level of the NLRP3 protein in this particular case here. On the left side in the same study, they stimulated the cells with lipopolysaccharide and A beta, interesting combination, and they found that the NLRP3 in the healthy controls went up by a factor of 3, 2.9, and it went up by 1,334 in the Alzheimer's cells. So we're not talking about a small difference here. That's about a 300 or 400 fold difference between the controls and the full-blown Alzheimer's here. And some of the other proteins showed a similar increase. So basically, Alzheimer's cells are primed to respond, and they respond very robustly when stimulated for the inflammasome response here. Another study measured the levels of cleaved caspase 1 here. Again, in the lower left is this blot, and you can see alternatively the control and the Alzheimer's matched ones, and the Alzheimer's ones have a higher level of the cleaved caspase 1 in the frontal cortex or in the hippocampus, more pronounced in the frontal cortex. And this again indicates activity is increased because the caspase 1 cleavage is an indicator of activity here that's elevated in the Alzheimer's patients. And in the Alzheimer's mouse models here, Morris water maze study is shown in the upper right. And when the animals are put into this water maze and they've learned where the platform is, they typically find it in the quadrant 1 over here. But if they are the Alzheimer's mice, they just keep looking and they don't remember where it was. But what was interesting in this study, they had knockout animals where they knocked out the NLRP3. And when they knocked out the NLRP3, the Alzheimer's mice did fine. They found the platform readily, as if their cognitive function, that was the conclusion, was actually maintained, despite the fact that they had the APP and PS overexpression here. So again, I'm hoping I can convince you that there's a link between the NLRP3 and Alzheimer's that way. And then let's turn our attention to connecting the dots here. And that is, what is it about the pathways that have to do with transsulfuration and methylation that impinge on the NLRP3 and that might explain the higher activity in Alzheimer's? Well, if you're looking for something to do with oxidative stress affecting proteins, I suggest looking for what the cysteines are doing, because it's the cysteines typically that are sensitive to oxidative stress. They either form oxidized disulfide bonds or they're reduced. So this is the point that I took, at least in thinking about this, and what we're going to look at progressively here is ideas and sort of some facts about how it is that the cysteine residues in the NLRP3 might be involved in its regulation by oxidative stress. On the right side here in green is shown again the NLRP3 molecule, at least most of it. In blue, again, is that NEK7. It's a kinase, NEK7, that gets bound by the NLRP3, and its binding is necessary for activation of the inflammasome. So, and this NEK7 is interesting because it's involved in cell division. And when it's sequestered by the NLRP3 in, let's say, an inflamed cell, for example, that sequestering of the NEK7 means that that cell is not going to divide as much. It inhibits cell division. And this is a feature of inflamed cell and inflammation in general. Cells don't divide as much when they're undergoing inflammation. This seems to be a part of the inflammatory response. So it's interesting that this is bound by the NLRP3, and it seems to play an important role here. So we started to think about the cysteines in the NLRP3, and there are some X-ray crystal structures here of the pyrin domain of the NLRP3 on the left, for example, here. And in there, they found a disulfide bond when they looked at the pyrin domain. And in this particular study, they speculated that, well, maybe that disulfide bond might be regulating the NLRP3, and that's the thought that I like and that we'll pursue here. And you can also have intermolecular sulfide disulfide bonds between two different molecules, as was demonstrated here for the NLRP12. So we started to think along these lines here, which some of this is hypothesis. I have to say, this is thinking, and it's a sort of a journal club here. Right? We're talking about what the literature suggests, more or less. And if you'll allow me to talk in that way, I'm going to suggest that the status of cysteine residues in the NLRP3, either being oxidized as a disulfide bond or being reduced as a thiol or reduced thiol, that determines the state of activity of the inflammasome, and therefore the inflammation state or the probability of inflammation. So here, I'm really saying that redox status of these cysteine residues determines the inflammation prospects here. And this is reversible. We know that there are certain proteins like thioredoxin that can reduce a oxidized disulfide bond. It does that using reducing equivalents that come through this selenoprotein thioredoxin reductase from NADPH. And interestingly, and probably importantly here, the ability of the thioredoxin to reduce disulfide bonds in NLRP3 is regulated by a protein called TXNIP. And TXNIP is thioredoxin interacting protein. And it's substantial literature that the TXNIP, when its levels are high, it interferes with this thioredoxin reduction, resulting in the NLRP3 being more active, associated with having more oxidized disulfide bonds that way. There's more to the TXNIP story. Its activity is increased in metabolic syndrome associated with diabetes and things like that. So it provides a connection between those features, metabolic features, and inflammation, if you will. So the TXNIP is an important part of the story here. And so the once the reduction, or if the NLRP3 is in its reduced state where the thiols are reduced here, it can be further modified by the addition of hydrogen sulfide. This is called either persulfidation or sulfhydration. But when this occurs, then you have, instead of just having a single SH, you now have like a protective group added to that, another SH added to it as a persulfide. And this further stabilizes, if you will, the inactive reduced state of the inflammasome. And so this is going to have a protective effect, a neuroprotective effect, if we're thinking about the brain and about Alzheimer's disease. Of course, for this to happen, you have to have enough hydrogen sulfide. And as we just said, it looks like the Alzheimer's brain has less than it should. And if that's the case, then this role of hydrogen sulfide persulfidation might not be what it should be, leading to increased level of inflammasome activity here. And there's plenty of studies out there showing that hydrogen sulfide inhibits the NLRP3 activity out there by different people. And even in COVID, there's certainly evidence that people that are low in hydrogen sulfide are at higher risk of mortality from COVID infections here. And a similar modification, not involving hydrogen sulfide, but involving oxidized glutathione can also occur here. This is called glutathionylation. And so the oxidized glutathione can bind to the SH group, and that actually releases a GSH when that happens. And this is another modification that will stabilize the inactive conformation here, because it disallows formation of a disulfide bond. So you can see that on the bottom here, we're accumulating different things to inhibit inflammasome activity, but they depend upon having enough glutathione around concentration-wise, or enough hydrogen sulfide around to accomplish that. So in the last year or so, there's been a couple of interesting papers that I've just noted here, both of whom looked at the glutathionylation. And what they found was the NEC7 protein itself has a cysteine that gets glutathionylated, and it needs to be removed in order for the inflammasome to be active. Likewise, that ASC protein that I mentioned that's involved in forming the complex, that also gets glutathionylated at a particular cysteine, and its removal of that glutathione leads to increased inflammasome activity. So what we can appreciate from these is that the thiols in various proteins, not only the NLRP3, but its partner proteins that it combines with, are all regulated by glutathionylation. And this, when they're glutathionylated, it interferes with activity of the inflammasome and tends to dampen it. And but when the glutathione is not on there, it increases the activity of the inflammasome. And here a colleague and friend in Japan, Chanli Zhang, he published an interesting study last year where he showed that when you stimulate cells with ATP, in this case as a stimulus, extracellular ATP, the intracellular glutathione just drops like a rock. It goes down here. You can see it's just almost disappears. And this is associated with activation of the inflammasome, as shown by the IL-1 beta level going up here. So this means that when you take away the glutathione from the inside of the cell, and you can no longer glutathionylate things as efficiently, that you're going to get increased activity of the inflammasome. So we can see how the glutathione availability and status makes a difference and is part of the activation mechanism. So I tried to just draw a little cartoon to capture what I'm saying here, and that is on the left, that if you got reduced proteins here, maybe the NLRP3 itself and or its combination with NEC7, or that ASC protein as another partner, if they're reduced, they stay separate from each other. But if they get oxidized or they're not glutathionylated, they can form this sort of network of oligomers on the right that can lead to speck formation and can sustain the inflammasome at a higher level of activity here. So this is, a sort of simple-minded, but nonetheless, I think it's a reasonable view of how the redox status of thiols regulates inflammation here. Now, another aspect, we all sort of interested in the coronavirus in particular. And it came from Wuhan in that area, and I suppose it came from bats, although there's a lot of debate about that. I noticed this particular paper, which came out about a year before the start of the pandemic. It came out of the Wuhan virology labs over there. And what they looked at here, they compared the response of the NLRP3 inflammasome from bats or humans or mice. And on the lower left over here, we can see the PBMC response to stimulation in humans is rather large. It's the open columns here compared to the red columns, which are the bats. So this really implies that the bats don't seem to respond and therefore are protected against the inflammatory response of the NLRP3 activation here compared to humans. On the right, we can see kind of a similar thing for mice, that the mice respond more robustly, whereas again, the bats don't respond very well at all. And in this paper, they tried to track it down a little bit at the level of the protein to say what could be responsible for this difference in sensitivity. And when they looked here, you can see in the upper right, the LRR region, the leucine-rich region, turned out to be the area that made the difference. Because when they created chimeric proteins here that had the human sequence here, that it had a higher response. And when they had the bat sequence of the LRR, they had a lower response here. So this implied that somewhere in that leucine-rich repeat was responsible for the difference in sensitivity between the species here. So I thought that was interesting, and I think it is interesting. So I thought I'd follow that up. And so I did a little sequence comparison here I'll share with you today of the bat, human, and mouse sequences aligned here for the LRR region here. So this starts about 700 in the amino acid sequence. And what I did is I color coded the cysteines here. And if you'll stay with me here at the bottom here, if a cysteine was conserved and it was a single cysteine by itself, and it was conserved for all three species, I made it purple. If it was a single cysteine that wasn't conserved and varied, I made it the aqua color here. And you see there's a fair amount of purple here. The purple ones indicate that these cysteine residues are conserved between the three species. And I did the similar thing for what's called vicinal cysteines. When you have two cysteines right next to each other, those two cysteines can form right next to each other in a disulfide bond called a vicinal disulfide. Interestingly, the NLRP3 has 6 of those overall, and 5 of them are actually in the LRR region here. So the vicinal cysteines that are conserved, I labeled green. You can see here 1, 2, 3, 4. 4 of them are conserved, and there's one down here in yellow that's not conserved. That it's a little, it's not a double cysteine in mouse. So in any case, this indicates overall that there's a good conservation of these cysteines between these 3 species. The difference that stood out between them that might be accountable for the sensitivity of the bats being lower is that the bat sequence is longer. It has an extension down the bottom. It actually goes a little further here, but there's no more cysteines. And the extended bat sequence has some extra cysteines in it. And I can speculate personally here that that might have to do with the lower sensitivity of the bat to the activation of the NLRP3 in terms of inflammation. But what I noticed even more, and you can see this from what I drew here, are these red lines here, which connected because I noticed a pattern among the cysteines. You see how this one, this one, this one, this one, and this one, they align to form sort of a straight line. Just, oh, these align here too. This number 1, 2, 3, and they look like they're the continuation of the ones on the left, which are earlier in the sequence there. And so there's another alignment here, number 2 and number 3, and they're parallel to each other. And what that implies when they're parallel is they're separated by about the same number of amino acids in the sequence. Turns out to be about 70 amino acids that separate these cysteines, and that they're sequentially positioned just 70 amino acids from each other. So if we look at the protein itself in three-dimensional form here, I just took this from a molecular graphics version, we can see that those lines that connect the cysteines, there's line 1, which is a continuation of the other one, and then line 2, 3, and 4, that they are associated with a position of those cysteines at a shared location. They, for instance, all the ones in line 2 are at the top edge of the leucine-rich repeat here. All those in line 1 are at the bottom edge. And the way they're positioned, that 70 amino acids, means that it's every other helix here has a cysteine. Not every one, but every other one, because it's the 70 amino acids that it takes to go up and then down and back again to reach the next cysteine level here. So what I'm trying to convey here is this kind of, I think, remarkable sequence at the amino acid level shows itself at the level of the protein as a repeating presence of these cysteine residues at specified locations along the outer portion of the leucine-rich repeat here. And I don't know exactly what this infers function-wise, but we're going to look into that. But it seems to me as though this might be involved, these cysteines might be involved in maybe one of the monomers binding to another one, or perhaps binding to the ASC protein or some other component of the inflammasome. And we're going to have to look into that. So, but otherwise, I just want you to appreciate that looks like the cysteines are pretty interesting in this NLRP3 protein here. So the last little bit I want to say here has to do with, okay, if that's all true, what does it mean for Alzheimer's treatment? We know that there's a crying need for treatment of Alzheimer's disease, and that pharma has not really been able to deliver the goods in terms of drug therapy here. And I'm going to start off by just saying, it looks to me like Alzheimer's is a dysfunctional aging associated with metabolic dysfunction of these pathways here. So if that's the case, then why not use metabolic treatments? I can say part of the reason is because you can't make $1 billion off of them, but are they effective? If true, we should see some effectiveness of treatments, metabolic treatments, I'll call them, nutraceutical treatments that are targeted against these pathways. And this goes along with the fact that it looks like the risk, according to at least this study, again from Sweden, was that the higher blood levels of vitamin B12 were associated with a lower rate of gray matter loss here. So there is evidence even from that kind of a study, that it's good to have higher levels of vitamin B12. And yeah, there was this study here that was done in, well, I guess they reported the results in 2015 or something like that by David Smith, Helga Refsum, and colleagues over at in Oxford in England, where they just treated people with, I think it was MCI definition here, and followed them over time and treated them either with a combination of these three vitamins here: B12, again, the cofactor for methionine synthase, folate that provides the methyl groups, and vitamin B6 or pyridoxine, pyridoxal phosphate, which is the cofactor for the two transsulfuration enzymes. So this is a good combination of things. If you're going to give vitamins, then these are a good combination. And what they found was that the rate of atrophy, gray matter loss, was sort of leveled off by this treatment as compared to the case for the placebo treatment during that same period of time. They could do this with some MRI structural analysis, and the yellow on the left over here was the amount of loss in the placebo on the upper level, and the lower level was the amount in the B vitamin supplement group, and it was less gray matter loss under that condition. And on the right, they looked at the tertiles of homocysteine here, and those that came into the study with the highest levels of homocysteine were the ones that had the most benefit from the B vitamin regimen that they used here, which again is not surprising. It just points to the role that these vitamins play in these sulfur pathways here. There was another study more recently reported. They used just folic acid and B12 without the B6 for a 6 months treatment here, evaluated cognitive function and some other endpoints, and found a significant improvement in the treatment group here, again, with folate and B12 being administered. What was also interesting here was at the top, they just documented that the blood levels of the metabolites improved, if you will, along with the blood levels of the B12 folate. But at the bottom here in this figure, they also showed that the TNF alpha levels went down with this combination of B12 and folate, which is kind of quite remarkable, suggesting that somehow activation of the methylation pathway or support of the methylation pathway is related to the amount of TNF alpha that gets produced. So in terms of other possible treatments, I'm not a clinician, I'm a bench research guy, but we can see from what I've talked about here, it would make sense you would want enough sulfur resources. You could get that by N-acetylcysteine or from whey proteins, a rich source of cysteine as well. But if you're aware of it, it turns out in the United States, the FDA is moving and has moved to take N-acetylcysteine off of the available nutritional supplement list. So the other day I tried to order some N-acetylcysteine. I couldn't order it. It's not being sold anymore because of this FDA action. I think it's particularly ill-timed to do that. But if we could get N-acetylcysteine, it would inhibit, and that's what's shown on the right here, it would inhibit the NLRP3 activity for as much as it could provide, for example, hydrogen sulfide to quench the activity of the inflammasome. And another interesting thing here, sort of lastly, is good old donepezil, which of course is a mainstay for treating Alzheimer's. It is a cholinesterase inhibitor, but as it turns out, it was just recently reported, this also has an inhibitory action on the NLRP3 inflammasome, as it turns out. we can see here in this part of the figure, here's the NLRP3 protein here, Western blot. And in the middle, we can see that the LPS stimulated it. This is in microglial cells. But when they treated it with this relatively high concentration, but nonetheless high concentration of donepezil, that it really remarkably decreased the NLRP3 level as a result of that. And this was associated with less of the formation of the IL-1 beta here in the lower right as well. So who knows, maybe, and I never really understood exactly why the donepezil would, as an anticholinergic drug, be even as effective as it is, which is of course limited, but maybe it has to do with this activity of inhibiting the NLRP3. So let me just wrap up by saying in conclusion here that the NLRP3 inflammation, which we can think of as neuroinflammation, it normally increases with aging. There's always an increase with inflammatory diseases with old age. You could call that inflammaging, for example. But it appears as though this is more extreme and hyperactive in Alzheimer's disease patients here. And this may have to do with the status of the cysteine residues in the NLRP3 inflammasome, because they seem to be an important determinant of that inflammatory activity. And then finally, I think there's a time based on that we should consider and look for treatments, especially low cost, but effective treatments that can address what is really a crisis in terms of Alzheimer's disease in the United States and elsewhere. And finally, let me just acknowledge the collaborators and people that have been in the lab and worked with us, especially Dr. Trivedi, Mala Trivedi, who's been a colleague and continues to be a colleague collaborator here at NOVA. Here, Marion Lemley, who was involved in some hydrogen sulfide studies and certainly had a lot of good exchanges with Marion about the underlying role of hydrogen sulfide here. Graduate students that produced some of the results I shared. And those are some who are still in the lab. Dr. Schreier, Matt, having just finished his PhD about a month ago here. And Janan Dong, who has done a lot of the mass spec work that we're doing for thiol analysis. So let me stop here if I could, stop sharing and just stop talking. And hopefully we have some time here, I guess, but I've gone over. But any questions, I'd be happy to answer some.
Dr. Deth, thank you for that. That was really clear and informative. And some of those observations towards the end of your talk were so intriguing and very interesting on your part to make that sort of follow-up. So thank you for presenting today. We are over time, but maybe we have time for one or two quick questions. I myself have to leave at 1:15. So I think to make this quick, please just unmute yourself and ask Dr. Deth a question and try to make it shorter than longer, since we really do only have a couple of minutes.
Sorry, I used up all the time.
That's okay. It was really worth it. I mean, I really enjoyed it. So who wants to speak up first? I have my own questions, but I'm going to give, I can ask Dr. Deth these questions anytime. So if anyone from the audience has a question, I encourage you to speak up.
Hi, Dr. Richard. It was really lovely presentation, and I see a lot of similarity between your work and Dr. Albensi's work in terms of NF-kappa B pathway involving in the inflammasome regulation and all these things. Frankly speaking, I have a lot of questions, but I'll stick to one particular question. You did a lot of analysis of postmortem human tissue samples for methionine synthase expression. And you shown some data showing the difference in splicing pattern in the Alzheimer brain, which you think associated with the enzymatic activity. Now, I mean, the postmortem human tissue is kind of tricky because of the RNA degradation. So did you see any, did you follow up with the protein expression to see whether the difference in RNA expression is kind of due to postmortem
Well,
intervals?
in one case, one important case, I didn't take time to talk about it. We did for methionine synthase in particular. We saw that 400-fold decrease.
Right.
We said, oh, does that mean that the methionine synthase protein is going down by 400-fold? No. In fact, the methionine synthase protein level stayed approximately the same. And that was a head scratcher for quite a while until we appreciated that this may represent a special situation for methionine status to control turnover of proteins and things like that. Because methionine, of course, is the first codon in all proteins.
Right.
And as such, it gets to call a tune for how fast a protein will be made through translation. If you will. So in any case, the protein stayed the same in that case, but we had to come up with a creative explanation for why that was the case. We haven't measured the protein for the other things that I showed, like NLRP3. We haven't done that yet. It's somebody's thesis project to do that. But the one we did measure was kind of intriguing in that way.
Because one
I'll
thing
just remind everyone, sorry, I'll just remind everyone that this is being recorded today. If you follow me on LinkedIn or Twitter, I'll post that link and it'll be on YouTube. So you can also do a search. Dr. Deth's name, my name, Nova, and the name of this presentation today. So you can find that recording. So maybe just to pass it on to someone else. If someone else has a very short question, we have maybe 1 or 2 minutes. Sorry, Sabir.
Just a quick question here. I'm glad that you brought up thyrodoxine. We are interested in thyrodoxine and follow that up, and we are seeing really interesting results in case of upstream of NLRP3 activation, and hopefully down the road we will be able to talk to each other more. So any information on the glutathione reductase or peroxidase changes in Alzheimer or aging?
Not that I'm aware of those two. There's a lot of players to think about here, glutaredoxin itself. It's interesting that when the inflammation is activated, that the cell kicks those ends, kicks glutaredoxin and thyrodoxin out of the cell
Yeah.
in order to create that oxidative environment inside of the cell.
Yes.
But thanks. I look forward to talking about the thyroid, about the TXNP story in particular, because the gene is regulated by methylation.
Yes.
And so that gives a feedback loop.
I have to look into that one. Thank you very much.
Thank you.
Dr. Deth, this is a monthly format. I think we're going to have you again in the spring to follow up on some of these questions, because I know that there's a lot of interest, but I think we're going to have to conclude right now. Thank you for your time today. I really appreciate it. And I'm going to have to follow up with you personally to ask you my questions. So
NF kappa B.
Yeah, there we go. Yeah, that's right. So thank you for coming, and look for our announcements in the future for the November Journal Club. Thank you very much.
Thank you.
Bye-bye.